All right, welcome back to the Deep Dive. Today we're going to be taking a look at something pretty interesting.
Yeah.
I hope so anyway. It's a microbe, and it's got some pretty out-of-this-world origins.
Yeah.
Literally.
Yeah.
It's called Kalamiella piersonii.
That's right.
And you guys have sent in some pretty fascinating research on this. And we're going to try to, break it down for everybody. So we're going to be talking about this bacterium, Kalamiella piersonii, and how it kind of went from, being found on the International Space Station to some pretty unexpected connections to human health.
That's exactly right. We've got scientific papers about how it was discovered, what makes it unique, and most importantly, we're going to look at some recent findings that link it to human infections. And we're going to synthesize all this for you, really focusing on the key takeaways and trying to keep things clear.
Yeah, we're going to try to keep it, away from the, too in the weeds stuff. So, okay, this is pretty fascinating.
Yeah.
The story of Kalamiella piersonii begins, in a remarkable place, the International Space Station.
That's a pretty cool origin story.
Right. Back in 2019, a team of scientists, Singh and his colleagues, at the Jet Propulsion Laboratory and the California Institute of Technology, they identified this bacterium. And their work, was published in 2019. You guys sent us the article.
Right.
And there's some, extra information online that kind of supplements it as well, that gives us more detail.
Right.
I mean, a microbe, that has ties to space.
Yeah.
I mean, that's a pretty cool beginning.
Absolutely. And it's really fascinating to see how something that was discovered in such an extreme environment could be linked to human health. And actually, there's a little side note here. Even back when it was first identified, there was a minor scientific correction that had to be made. There was a follow-up article, also from 2019, that clarified a mistake in the original paper's taxonomic description. Turns out there's this thing called the International Code of Nomenclature of Prokaryotes.
Oh, okay.
Which are rules about how you name things, and it says you have to describe a new genus separately from a new species within it.
Got it.
It's important for scientific accuracy.
Right. Got to keep it all straight, I guess.
Exactly.
Okay, so let's dig into this bacterium a little more. So the genus, Kalamiella, was actually named to honor APJ Abdul Kalam, who was a really, really important Indian scientist who made huge contributions to space research.
That's right.
Which I think is so fitting, given where this microbe was first discovered.
It is, yeah. It's a nice tribute.
So now let's talk about what makes the Kalamiella genus special. What are its defining characteristics?
So Kalamiella bacteria are what we call gram-stain negative. Basically, that means their cell wall has a thinner outer layer than some other bacteria.
Okay.
They're also aerobic, so they need oxygen to survive and grow. And they're motile, meaning they can move around on their own.
Interesting.
If you look at them under a microscope, they're short, rod-shaped cells, usually found alone or in pairs. And when they're grown on a standard lab medium, something called tryptic soy agar, or TSA, at 30 degrees Celsius for 24 hours, their colonies have a specific look. Circular, slightly raised, beige color.
Okay.
And they're usually about 0.6 to 1.0 millimeters in diameter.
So we're starting to get a visual here.
Yeah, exactly.
So how about the conditions that they can tolerate? Are they picky about where they live?
They're actually pretty adaptable. They can grow in a temperature range of 12 to 37 degrees Celsius, but they seem to thrive best around 30 degrees.
Okay.
They also have a pretty wide pH tolerance, from slightly acidic at 6.0 to alkaline conditions up to 10.0.
Oh, wow.
So that tells us they can survive in a lot of different environments.
Yeah.
They can also handle salt concentrations from 0 to 5% sodium chloride. Now, on a molecular level, they have specific fatty acids in their cell membranes, mostly C16.0, C17.0 cyclo, and then these things called summed feature 3 and summed feature 8. These are just specific combinations of fatty acids.
Okay.
And the last thing is that their DNA has a guanine and cytosine content of 57.07%.
Got it. So that gives us a pretty good basic understanding of the Kalamiella genus.
Yeah.
Now let's zoom in on Kalamiella piersonii, which you said is the representative species for this entire genus.
Right.
And this species is actually named after Dwayne Pearson.
Yeah.
Who's a well-respected American space microbiologist.
Yeah, he is.
So what makes Kalamiella piersonii stand out from the rest of the Kalamiella genus?
Well, in addition to the general traits we talked about, Kalamiella piersonii cells are about 1 to 1.2 micrometers wide and 2.8 micrometers long.
Yeah.
And while the genus can grow in a pretty broad temperature range, K. piersonii is a little more limited. It doesn't grow at really cold temperatures like 4 degrees Celsius or really hot temperatures like 44 degrees Celsius. It still grows best at 30 degrees. And even though the genus can tolerate a wide pH range, K. piersonii prefers things a bit more alkaline, ideally around 8.0.
Got it.
Now, the 2019 correction article goes into a lot of detail about the different carbon sources that K. piersonii can use. Basically, these are things like sugars and amino acids and other compounds that it uses for food. For instance, it can use things like glucose and fructose and asparagine, but it can't use trehalose or sucrose or deserine. So all this information about what it can and can't use for food is super helpful for scientists who are trying to accurately identify it.
Right, because it's all about, getting those specific details.
Absolutely.
Okay, so now we have a pretty good picture of what Kalamiella piersonii is and where it was first discovered. But now things are going to get pretty interesting.
Yeah, here's where things take a turn.
Because this is where the story takes an unexpected turn. And this is where it gets really, really interesting.
Right.
You mentioned before, this connection to human health.
Right.
So this is where that 2021 paper by Rekha and his colleagues.
Yes.
This is where that paper really seems to be a pivotal moment in, this whole story.
Absolutely.
So tell us about that discovery.
So this 2021 paper was the first time that anyone had ever found K. piersonii in a human. What happened was researchers identified a strain of it. They called it YU22, and it's also sometimes called MCC3182.
Okay.
And they found this in the urine of a patient who had a struvite kidney stone.
Wow.
Now, when they first discovered the strain on the ISS, computer analysis suggested that it wasn't likely to colonize humans.
Right.
And there were no previous reports of it being found in people at all. So this was pretty significant. And what's interesting is that they initially misidentified the YU22 strain as Pantoa species.
Oh, really?
Which just goes to show how tricky it can be to accurately identify this bacterium.
Right. So that could be a problem then.
It can be, yeah.
So this is a big leap, though, from finding it on the International Space Station to finding it
hugely
in a kidney stone patient. So what was it about this YU22 strain that made the researchers think that it could actually be related to issues with human health?
So the researchers dug deep into the genes and functions of this YU22 strain. And what they found were several traits that are classic signs of bacterial virulence.
Okay, so what do you mean by virulence factors?
Virulence factors are basically the tools that bacteria use to do a few things. Establish themselves inside a host, get around the host's defenses, and sometimes even cause disease.
Gotcha.
It's like their arsenal for causing trouble.
Okay, so let's talk about some of these tools, or some of the important ones they found.
Well, the YU22 strain has genes for things called adherence factors, which are like molecules that help the bacteria stick to host cells.
Okay.
Sort of like Velcro.
Gotcha.
It's also got a full set of genes for chemotaxis and motility. Now, chemotaxis is like a sense of direction for bacteria. It lets them move towards things that are good for them and away from things that could harm them. And motility is just the general ability to move around.
Right.
The genes involved in these processes are called She, Mot, Fel-G, Fel-AH, and Fly.
Got it.
They also found genes for type-fall pilu, which are these hair-like structures that help with attachment. And they found a Fimbriae-like adhesin called SFMA. So all these things suggest that this bacterium is very good at moving around.
Right.
Which is probably important for that initial attachment and then being able to move into host tissues.
So it's got the tools to get around and get where it needs to go?
Exactly.
Now, was this ability to move something they just predicted from its genes, or did they actually see it happening in the lab?
They actually put it to the test. They did experiments and found that the YU22 strain was highly motile.
Really?
Both in liquids and on these semi-solid agar plates they use in the lab. When they did a swimming assay, the bacterial colony spread out to a diameter of 30 millimeters in just 24 hours.
Wow, that's pretty fast.
Yeah, it is.
So it can really get around.
It can. They also looked at something called swarming motility, which is a different type of movement on surfaces, and that was a bit slower, but still noticeable over time. And they also saw that it could form biofilms.
Now, remind me what biofilms are again.
Biofilms are these communities of bacteria that group together and surround themselves with this protective matrix.
Okay, so it's like they're building a little fortress.
Exactly, yeah. It helps them survive.
Right.
They saw these biofilms forming on plastic surfaces, even in conditions where there wasn't much movement. And biofilms are often linked to infections that are really hard to get rid of.
Right, because they're kind of protected in there.
Right.
So this is not good news.
It can be a problem, yeah.
So they were able to actually see these things happening in the lab.
Oh, yeah, they went even further. They used scanning electron microscopy to get a really close look at the bacteria.
Wow.
And they found that it has what's called Lophotrichous flagella.
Okay, break that down for me.
So flagella are those whip-like structures that bacteria use to move around.
Right.
And Lophotrichous means that there are multiple flagella, all located at one end of the cell.
Interesting.
Yeah, and this arrangement is often associated with really good swimming ability.
So it's like it's got a powerful little motor on one end?
Exactly, yeah. And they also discovered that YU-22 can produce cytophores.
Okay, and what are those?
Cytophores are molecules that bacteria use to capture iron, which is a really important nutrient that's often hard to come by inside a host. They were able to confirm this by using something called a CAS-agar assay, which is a specific lab test.
Okay.
It involves a color change that indicates the presence of cytophores.
Got it. So it's equipped to move around, stick to things, form those protective communities, and even scavenge for these essential nutrients.
Yep.
It sounds like it's really well prepared to be a pathogen.
That's right. But there's more. They also tested how well it could survive against the host's immune system.
Oh, okay. How did that go?
They found that YU-22 showed some resistance to being killed by serum, which is the liquid part of blood. When they exposed it to healthy human serum, they saw about a 20% reduction in the number of bacteria.
Okay, so not completely resistant?
No, but still some resistance.
Right.
They also wanted to see how it interacted with human kidney cells.
Yeah, that makes sense given the kidney stone connection.
Exactly. They use a specific type of kidney cell called HEK293T.
Okay.
And they saw that YU-22 was really good at sticking to these cells, with around 46.5% of the bacteria attaching.
Wow. So almost half.
Yeah. And what was even more concerning is that they saw a lot of damage to the kidney cells. Around 67% of the cells were damaged.
That's a lot.
Yeah, it is. And they even saw signs of apoptosis, which is basically programmed cell death.
So this bacterium isn't just hanging around.
No.
It's actually causing harm.
That's what it looks like, yeah.
Okay, now let's talk about how it deals with urea, because we're talking about the urinary tract.
Right.
The paper talks about that too, so what do they find?
So this is where things get a little different.
Huh.
Unlike a lot of bacteria that cause urinary tract infections,
Yeah.
YU-22 doesn't actually have the gene for urease. Now, urease is an enzyme that a lot of those bacteria use to break down urea into ammonia.
Right.
But they did discover that YU-22 has another way of breaking down urea.
Okay.
It uses two enzymes called urea carboxylase, which is shortened to UC, and alatinate hydrolase, or AH.
Got it.
And this system is similar to what they found in the original ISS strain.
Interesting.
So by looking at the genetic relationships, they found that the urea carboxylase in YU-22 is pretty similar to urea amyloidases, which are enzymes found in fungi. And the alatinate hydrolase kind of forms its own distinct group within a family of bacteria called Erwiniaceae.
Got it. That's a lot of complicated names.
Yeah, it can be a mouthful. The main thing to remember is that this pathway with the UC and AH requires a vitamin called biotin to work.
Okay, so it's found a different way to deal with urea.
Yeah.
What are the implications of that?
So they also looked at how YU-22 uses amino acids, because those are found in urine too.
Right.
And they found that it can actually use amino acids like asparagine and glutamine as its only source of carbon.
So it's resourceful.
It is. But it did grow a bit slower when it only had those amino acids compared to when it had glucose available.
Okay, so glucose is still the preferred fuel source.
Seems like it, yeah.
But what's really interesting is that when YU-22 used these amino acids, it made the growth medium much more alkaline.
Okay.
Compared to when it was growing on glucose.
So the pH went up.
Exactly. Right. And they confirmed that it has the enzymes asparaginase and glutaminase, which help break down those amino acids.
Okay, so that explains the pH change.
Yeah.
Now, how does this pH increase tie back into the whole kidney stone issue we were talking about earlier?
That's a great question. This is a really crucial connection.
Okay.
So when YU-22 breaks down those amino acids,
it releases ammonia, which makes things more alkaline.
Right.
And just like what happens with bacteria that produce urease, this release of ammonia can contribute to the formation of struvite crystals in the urinary tract.
So it's not just the presence of urine that can lead to those crystals.
Right.
Now, they actually did this in the lab.
They did.
They did experiments using artificial urine, and they actually saw crystals forming around the YU22 bacteria.
That's right.
Wow.
And they took a closer look at these crystals using a scanning electron microscope.
Okay.
And they found that they had these intricate structures. And they did another analysis using something called FTIR spectroscopy.
Yeah.
And that confirmed that they were indeed struvite crystals. And they were very similar to the crystals that formed in the control experiments.
Okay.
Where they just added ammonia directly.
Got it. So even without the usual urine enzyme, this bacterium has a way of making those crystals.
It seems so, yeah.
That's pretty fascinating.
Yeah.
Now, one of the big concerns with any potential pathogen is its resistance to antibiotics.
Right.
So what did the researchers learn about YU22's resistance profile from that 2021 paper?
They did a genetic analysis and found several genes associated with antibiotic resistance in YU22.
Okay.
They found genes linked to resistance against polymyxins, which are a class of antibiotics that are often used as a last resort for really difficult infections.
So that's not good.
No, it's concerning.
And what else did they find?
They found genes for a beta-lactamase enzyme, which can break down beta-lactam antibiotics like penicillin.
Right, so that makes the bacteria resistant to those drugs.
Exactly. They also found a cluster of genes for resistance to fusaric acid. And they found genes for bacitracin resistance as well.
Okay, so it's resistant to a range of antibiotics already.
Yeah, it is.
And were there any other resistance mechanisms they found?
Yeah, they found several different efflux pump systems. These are like little pumps within the bacterial cell that can actively pump out things that are harmful to the bacterium, including antibiotics.
So it's like a defense mechanism.
Exactly. And this contributes to antibiotic resistance.
Right. Do you know which specific pumps they found?
They identified a bunch from different families like the RND, OMP, OMF, MFS, and MFP families.
Wow.
Specific examples include MDT-EBC Tulsi and achromity Tulsi.
It's like it's armed to the teeth.
It seems that way, yeah.
So all this is pointing to a potentially serious problem.
Yeah, definitely something to keep an eye on.
Now, the paper also included a table that showed the strain sensitivity to certain antibiotics.
It did, yeah.
So can you tell us a little more about that?
Yeah, so for example, they found that it was sensitive to levofloxacin and meropenem, but resistant to ampicillin and ceftazidime.
Okay, so a mixed bag, but definitely some resistance there.
Definitely.
So all this brings us to actual cases where K. piersonii has been found in human infections.
Right.
Now, we have some reports from 2022 and 2023, and even a poster from 2024 that detail these instances.
Yep.
So let's start with the 2022 case reported by Alpizar Rivas and his colleagues. What happened there?
All right, so in that case, they had a 56-year-old woman with a history of serious heart problems.
Okay.
And she was getting medication through a tunneled catheter.
Okay.
Now, she ended up developing chills, and the blood cultures came back positive for K. piersonii.
So they found it in her blood.
They did. Now, what's interesting is that the initial identification was really tricky. They used this technique called Maldi-Otho mass spectrometry, which identifies microbes based on their unique protein signatures. And that initially suggested that it was Pantoa glomerans, and then later they used more traditional methods, and those pointed to Salmonella.
It's a lot of confusion what this bacterium actually was.
Exactly. Even though it looked like Pantoa glomerans in the culture, it was only when they analyzed the 16S rRNA gene, which is a really common way to identify bacteria based on their genetic code, that they were absolutely sure that it was K. piersonii.
So the standard methods really struggled.
Yeah, they did.
To get it right, which is a really important point to highlight, I think.
Absolutely.
Because, if you don't identify the bacterium correctly, then you can't treat it correctly.
Exactly. It can lead to delays in treatment for sure.
So how did they treat this patient, and what ended up happening?
So they ended up treating her with a 14-day course of intravenous piperacillin tazobactam.
Okay.
And that was after they removed her tunneled catheter because they suspected that was the source of the infection.
Okay, that makes sense.
And thankfully, she recovered.
But it seems like they got it in time.
It seems like they did, yeah.
Okay, good. So what did the susceptibility testing show in this case?
So it showed that this particular K. piersonii isolate was sensitive to ciprofloxacin and moxifloxacin. So this case really showed that K. piersonii can act as what we call an opportunistic pathogen.
Right, meaning it takes advantage of a weakened immune system.
Exactly.
So in this case, it was someone with heart problems and a medical device that allowed the infection to take hold.
Yeah. And I think this really emphasizes how important it is to use more advanced methods like 16S rRNA sequencing to make sure we're identifying these bacteria correctly.
Yeah, it's really important to get the diagnosis right.
It is.
So now let's move on to the case from 2023 that was reported by Sada and his colleagues.
Okay.
Now, this one involved a much younger patient.
It did.
A 2-month-old baby girl.
Yeah, a very young patient.
So what happened there?
This case is particularly interesting because this little girl came in with some gastrointestinal problems like diarrhea, and she wasn't feeding well and she was vomiting. So initially, they thought she had something called acute enterocolitis, which is pretty common, but then she developed a fever, and a blood culture came back positive for bacteria.
So they knew it was something more serious than just enterocolitis.
Right. And again, the initial identification using MALDI-TOF MS was difficult.
So similar to that previous case.
Yeah, it is.
Sounds like this might be a recurring problem with this bacterium.
It could be, yeah. In this case, the MALDI-TOF suggested that it might be Pantoaseptica, but they weren't very confident about it. So they ended up analyzing the 16S rRNA, and they looked at some other genes called housekeeping genes.
Okay, what are those?
Housekeeping genes are essential genes that are pretty stable across different bacteria. And they're useful for identification. Some of the ones they looked at were GRRB, RPOB, and ATAPBD. And all of that confirmed that it was K. piersonii.
So again, it took some extra steps to get the right identification.
Yeah, it did.
So they were dealing with a 2-month-old baby with a blood infection. So how did they treat her?
They treated her with an antibiotic called cefotaxime. And thankfully, she recovered as well.
That's good to hear.
Yeah, it is.
Now, what about the potential sources of the infection? How did this baby get sick in the first place?
That's a good question. And in this case, it wasn't so clear. The researchers thought about a few possibilities. They considered that maybe bacteria had moved from her gut into her bloodstream.
Oh, right. That's called translocation.
Exactly. Bacterial translocation. They thought this might be related to a suspected food allergy. Specifically, a non-IG-mediated gastrointestinal food allergy.
Pretty specific.
Yeah, it is. And it's a type of allergy that was diagnosed later on. But they were careful to point out that there's no strong evidence to directly link this type of food allergy to bacterial translocation. So it's just a possibility they were considering.
So they weren't able to pinpoint exactly where the bacteria came from.
Not in this case, no.
This case is significant, right?
It is, yeah.
Because it shows that K. piersonii can cause serious infections, even in very young babies who don't have any obvious problems with their immune systems.
Right. It's not just affecting people who are already sick.
Right. So it's expanding our understanding of who might be vulnerable to this bacterium.
Absolutely.
Okay, now we have one more case to discuss. The 2024 poster by Abidin and his colleagues. Now, this one seems to be a completely different scenario from the other two.
It is, yeah. This one involved a 32-year-old man who had recently been diagnosed with advanced retroviral disease and hepatitis B.
So he was immunocompromised.
Yeah, definitely.
And what happened with this patient?
So this was the first time that anyone had isolated K. piersonii from a tissue sample. They took a biopsy from a skin lesion on this man. And that's where they found the bacteria.
Interesting.
Yeah, it is.
So how did they identify it this time?
So initially, they used MALDI-TOF MS again. And that gave them a log score of 2.23. But, of course, to be absolutely sure, they confirmed it with 16S RNA sequencing.
And what about antibiotic susceptibility testing? What did they learn about this particular strain?
They found that this isolate was producing an enzyme called AMPC, which makes it resistant to ampicillin and amoxicillin clavulinate.
So more antibiotic resistance.
Yeah, unfortunately.
But was it susceptible to anything?
Yeah, it was. They found it was susceptible to other antibiotics like cefepime and meropenem.
So there's still some options for treatment.
There are, yeah.
So what does this case tell us about K. piersonii?
I think it reinforces a couple of things. First, it seems like K. piersonii can be an opportunistic pathogen, especially in people with weakened immune systems.
Right.
And second, it highlights the fact that it can have multi-drug resistance.
Which is a really big concern.
Yeah, it is.
And also, this is the first time we're seeing it isolated from a skin lesion.
Yeah, that's right. That's a new finding.
So it seems like this bacterium can show up in a lot of different places in the body.
It does seem that way, yeah.